Every cell in your body has a copy of the same DNA. It started from an original copy and will copy itself trillions of times over the course of a lifetime, each time using half of the strand of the original template to build a new molecule. Enzymes unwind the DNA strand. After the enzyme helicase unwinds the double helix, it slices open the loose hydrogen bonds between the base pairs. Adenine and thymine pairings have 2 hydrogen bonds, making them weaker than the guanine and cytosine pairing, which have 3 hydrogen bonds. The splitting of the base pairs starts at the replication fork, which has a top strand, called the leading strand, and a bottom strand, called the lagging strand. Although both strands are complementary, they go in opposite directions. While the lagging strand goes from 5' to 3', the leading strand goes from 3' to 5'. The RNA primase attaches the primer to the starting point. This is so that the DNA polymerase can extend the RNA primer with complementary DNA. Now the parental strand has provided as a template for the new strand synthesis. Since the DNA polymerase can only copy from the 5' to 3' direction, making a leading strand is easier than making a lagging strand. Here you can see the RNA primase synthesizing a short RNA primer so that the DNA polymerase can extend this RNA primer with short lengths of complementary DNA. Since the polymerase can only copy strands from the 5' 3' direction, and the lagging strand is 3' 5' direction, the RNA primase has to lay down an occasional RNA primer that gives the polymerase a starting point, and so there are many segments along the stem. This leads to many individual segments, each 1,000 to 2,000 base pairs long, and each starting with the RNA primer, also known as Okazaki fragments. After the lagging strand has been replicated, the DNA polymerase I digests the RNA primer from the Okazaki fragments and replaces it with the corresponding DNA. And then the final enzyme, DNA ligase, joins together all the segments. James Watson and Francis Crick, who discovered DNA, concluded that it has not escaped our notice that the specific pairing that we have postulated immediately suggests a possible copying mechanism for the genetic material.